Hello friends, and welcome to the lecture series on edible vaccines. In my previous lecture, I discussed about some of the basics of our immunity with special reference to its types. And we categorize immunity on the basis of how it is acquired, that is active and passive immunity, or we also categorized it on the basis of its specificity, that is the humoral immunity and the cellular immunity. Now we discussed that humoral immunity is mainly carried out by the B cells, whereas the cellular immunity is carried out by the T cells. Then I gave a brief overview on the various components of humoral and cellular immunity and how these types of immunity work in our body. I also gave a view of what is mucosal immunity, what are the locations where it is located, like respiratory tract, gastrointestinal tract, urogenital tract, and skin is also part of the mucosal immunity. And this was followed by an overview on the components that are involved in GIT mucosal immunity. Now, since we are dealing with edible vaccines and edible vaccines are consumed through oral route, which eventually enters into your gastrointestinal tract, so I was mainly focusing on the GIT mucosal immunity. And in that we discussed the three main components of GIT mucosal immunity, that is epithelial barrier, which is having tight junctions, antimicrobial peptides, TLR receptors and NLR receptors, which form the first line of defense, followed by lamina propria region, which is the main connective tissue region, and the pair patches that are present in that particular region, and also the mesenteric lymph nodes, which was the other components of this system. So as I told you that mucosal immunity stimulates both innate as well as adaptive immunity. But the question is how the components of GIT mucosal immune system work in tandem to protect the body from the pathogenic microbes. And another question is that how these edible vaccines work. So friends, this is what I'll be talking about today. Now, in addition to this whole mechanism of action, I'll be just also giving you an overview of what kind of plants which we are using to prepare edible vaccines and what are the limitations or challenges for the edible vaccines. So friends, coming on first to that how this GIT mucosal immune system works. Now, as we discussed that lamina propria region and the subepithelial domain region, that is SED region, and we know that SED region lies between the follicular associated epithelial cells and pair patches. Now, both these regions have dendritic cells. So as soon as the pathogen or the harmful bacteria or the PAMPs, that is pathogen associated molecular patterns, are detected by the TLR receptors, they stimulate the secretion of chemokines like CCL21 or CCL19. Now these chemokines attract these dendritic cells because of the presence of certain receptors, namely CCR1 receptor and CCR6 receptor on the dendritic cell surface. Now when these cells or dendritic cells are attracted by these chemokines towards epithelial cells, they move toward the epithelial surface and capture the pathogen or the antigen or the PAMP. This is first mechanism. And second is, as I also told you in my previous lecture, that there are certain specialized cells which are present in the follicular associated epithelial region, and these cells were known as microfold or M cells. And these M cells are not covered by glycocalyx or the mucus coating. So these M cells also take up the pathogen or the antigenic molecule by phagocytosis, and then they again secrete those chemokines which attract these dendritic cells, and then they transfer that pathogen to the dendritic cell, which is present in the SED region, which is subepithelial domain region, and then these dendritic cells enter into pairs patches or they move on to the mesenteric lymph node. Now, in addition to these two mechanisms, that is TLR or M cell modulated response, dendritic cells can themselves extend their arm through the epithelial cell barrier into the lumen and they can sample the pathogen, or they can catch the pathogen or antigen and present to the macrophages. And when they present them to the macrophages for their destruction, this initiates the innate immune response. Or they can move. Some of the dendritic cells can also move to the lymphoid structures. And once once the pathogen is transferred to the dendritic cell, either with the help of TLR or M cells, or they capture on their own, they move toward lymph nodes or lymphoid structures, which include the mesenteric lymph node and the Peyer's patches. They stimulate the adaptive immune response, and that is why mucosal GIT has both innate and adaptive immune response. So as soon as the pathogen is transferred to dendritic cell, they upregulate the MHC class 2 response, that is major histocompatibility complex class 2 response, as well as they upregulate the expression of another kind of receptor, CCR7 receptor, that is also present on dendritic cells. Now when dendritic cells were attracted towards epithelial cell, this CCR7 receptor were downregulated, and once they capture the pathogen, these CCR7 receptor cells, they get upregulated and downregulate the CCR1 and CCR7 receptors, which took them to the epithelial cell. So this upregulation and the downregulation now allows the dendritic cells to move away from the epithelial cells and migrate towards the lymphoid structures, which are Peyer's patches or the mesenteric lymph nodes. And once the dendritic cells they engulf the pathogen or the antigen, they act as antigen presenting cell, because I already discussed this in my previous lecture, that there are certain cells which present antigen to the immune structures, and dendritic cell are one of the major and the most important antigen presenting cell. Apart from that, B lymphocytes are also considered as antigen presenting cell, but to a very small extent. And once these become antigen presenting cell, they present these pathogens or the antigenic molecules to the naive T lymphocytes in the lymph node to activate them. Now the question is that why dendritic cells present antigen or pathogenic microbe to T cells and not B cells? Now the answer lies in the structure of these lymphocytes. Now the T cell antigen receptors that are present on the surface of T cells are designed to sense or to see the antigen molecules displayed by the host cell surface molecules. This means that whenever a host cell will present the antigen, only then T cells can capture it or can sense it. And they will not sense the antigens which are present on the microbes or the antigens that are free in the circulation or the extracellular fluids. So in simple words, if I say that T cells can see the antigenic molecules or the PAMs that are presented or that are bound to the surface of the host cells, and these host cells can be dendritic cells or the infected host cells, but they cannot see those antigenic molecules which are circulating in the normal circulation or the extracellular fluid. Now this is the major difference from B lymphocyte, because B lymphocytes have certain antigenic receptors on their surface, and not only the B cells have those antigenic receptors, but the substances that are secreted by B lymphocytes, like antibodies, they can also recognize the antigen on the microbial surface, which is present in the circulation or in the extracellular fluid, or even sometimes some soluble antigenic proteins are also present in the circulation or extracellular fluid, which can be directly sensitized by the B lymphocytes. Or B lymphocytes, simple words, if I say, B lymphocytes can see only those antigenic molecules or the pathogenic bacteria which are present in the free circulation or in the extracellular fluids, whereas T cells will see only those antigens that are presented to them by the host cells, which are dendritic cells or the infected host cells. And therefore, dendritic cells present them only to T cells and not B cells. Now, since B cells have surface receptors that can recognize the antigens, so if any soluble antigen enters into the lymph node or Peyer's patches, that soluble antigen can directly stimulate the naive B cells. And we discussed that naive lymphocytes are those which have not been exposed to any kind of antigen. And this kind of direct activation of B cells or naive B cells is known as T-cell independent activation pathway of B cells. Now another question arises that how dendritic cells present antigen to the T cells. Now this is performed with the help of specialized protein, which, as we have already discussed, that there is an upregulation in the expression of MHC class 2 response. So T cells, dendritic cells, basically cause an upregulation of MHC molecules. What are these? Now there are two types of MHC, class 1 expression and MHC class 2 expression. Now class 1 expression is expressed by most of the nucleated cells, and its expression mainly deals with the killing action of the killer cells. So if activated T cells are present in lamina propria region, and if any pathogen enters or the infected cell is there, then MHC class 1 expression is upregulated, and those killer cells kill that particular infected cell, which is a sort of innate response. Whereas MHC class 2 response or the class 2 expression is done by dendritic cells, macrophages, and to small extent on the B cells. And this kind of expression mainly deals in the recognition of the antigen. And that is why whenever an antigen or a pathogen is transferred to dendritic cells, they upregulate the MHC class 2 expression and move towards the lymph nodes. So now dendritic cells carry the antigen either from M cell or directly or with the help of TLRs or NLRs, and now they enter into the pair patches or the lymph nodes and present them to the naive T cells. Now, this causes the activation of these naive T cells, and they get converted into helper T cells or CD4 cells. Now these CD4 cells further differentiate into killer or the effector T cells, which are also known as CD8 cells, and some memory T cells, along with some of the regulatory T cells, which will regulate the immune response later on. Now, in addition to this, these helper T cells or CD4 cells, they also activate naive B cells. And this activation process is known as T-cell dependent activation, which causes the activation of naive B cells. And these naive B cells then further differentiate and proliferate. Now, another question arises is that how helper T cells activate the naive B cells. Now the naive B cells have a protein which is the CD40 receptors on their surface, whereas helper T cells, they have an protein or a ligand which is known as CD40 ligand or CD154. So helper T cells express or upregulate the expression of CD154, and they also secrete certain chemokines. Now these chemokines, they basically move these helper T cells toward the follicular region in the pair patches, towards the germinal center of the pair patches, which we discussed in my previous lecture. So once they move toward pair patches, these CD154 ligand binds to CD40 receptor of the B cells. And once this ligation or binding happens, this causes activation of the naive B cells, which then further differentiate or proliferate. Now, once the B cells get activated, they differentiate into mature B cells or the plasma cells, which secrete antibodies. And apart from mature B cells or plasma cells, they also differentiate into memory B cells, which keeps the memory of the infection or the pathogen that has been introduced. Now all these activated cells now move toward the lymph node through the afferent lymph vessel, and apart from this mechanism, dendritic cells can directly enter into the mesenteric lymph node, thereby acting naive T cells in the lymph nodes, and these naive T cells then again get converted into helper T cells, which further causes differentiation of T cells into killer T cells or the regulatory T cells, and in addition to that, they again activate some naive B cells in the lymph nodes into mature B cells and the memory B cells. Though the B cells are now mature and they are producing antibodies, but still they are producing immunoglobulin M or the antibody M, and not antibody A or immunoglobulin A, which is the main antibody in gastrointestinal tract. So therefore, now the B cells undergo class switching process, that is, from secreting immunoglobulin M or antibody M, now they will switch on to a structure or to another class where they will now secrete immunoglobulin A or antibody A. Now this class switching is again assisted by the type 2 helper T cells. Now these helper T cells or the CD4 cells, they secrete transforming growth factor beta, which is TGF beta and interleukin 4. Now both these components, they help these mature B cells undergo class switching, and they start producing the antibodies A. Now in addition to TGF beta and interleukin 4, interleukin 5 and interleukin 6 are also secreted by these helper T cells, which help in maintaining the secretory B cells which secrete antibody A. Now ligation, as I told you that the naive B cell have a CD40 molecule, whereas naive T cells or the helper T cells, they have a CD154 ligand on their surface. So when this ligation or binding occurs, this also stimulates the conversion or the class switching of the B cells. Now once all these lymphocytes, they get activated, now they have to go to the lamina propria region to show their immune response. And for this, they have to leave the mesenteric lymph node and enter into the systemic circulation. Now, till now, whatever we have discussed, it exhibited the induction or stimulation of the immune response. It's not the effect of the immune response, but it's the stimulation of the immune response which has occurred in the lymph node or Peyer's patches. And therefore, these sites are normally known as inducer sites for the immune response. Now, majority of the action, almost all the action or the effect of these lymphocytes is exhibited in the lamina propria region, and therefore lamina propria region is considered as the effector site of these lymphocytes. Now once these lymphocytes or the activated lymphocytes enter into the systemic circulation, the question is that how they reach the lamina propria region. Because if something enters into systemic circulation, it can enter into any organ, any part of the body. Now, once the dendritic cells activate naive lymphocytes, that is T lymphocytes, another important process happened during this time. So these dendritic cells, they take vitamin A and convert it into retinoic acid. Now this retinoic acid induces the expression of a chemokine receptor, that is CCR9, and another protein known as integrin alpha 4 beta 7 on the surface of mature B cells or the plasma cells. Along with that, they also express this protein on the affected T cells also. Now these chemokine receptors and the integrin protein alpha 4 beta 7, they act as homing receptors, and they will guide these activated lymphocytes to the lamina propria region. Now what happens is the blood vessels or the capillary endothelial cells of the vessels in the lamina propria region, they have the ligands for the chemokine CCR9, and this ligand is known as CCL25, and another ligand which is MADCAM1, which act as ligand for the integrin protein alpha 4 beta 7. So whenever the activated lymphocytes with CCR9 and alpha 4 beta 7 integrin on their surface reach to lamina propria, these receptors bind to the ligands that are present in the blood vessels. So once they bind to those ligands, this bindation or ligation prevent the movement of these activated lymphocytes out of the lamina propria through systemic circulation. So they remain in that particular region, and then they diffuse out of those blood vessels, and once they enter into the LP region or the lamina propria region, they exhibit their effector action. That is, the T killer cells will kill the infected cells, infected host cells, and the mature B cells, they will secrete the antibody A, which neutralize the pathogens in that particular region, lamina propria region. And memory B cells, they retain the information about the antigen or the pathogen for the future action. Now once this antibody A is being produced by the plasma cells, this has to be transferred into the lumen also in order to neutralize the antigen that is present in the lumen. And before we move on to its transportation, first I will just give you a brief overview about the structure of immunoglobulin A or the antibody A. Now antibody A occur in the form of dimers, and the two monomers are attached together by the J chain through the disulfide bonds. Now this J chain is important, and I will not go further into the detail into the heavy chain structures or the light chain structure of this antibody. But the important thing is these dimeric antibody A molecules, they cannot cross the epithelial barrier on their own. And for this, they bind to a polymeric immunoglobulin receptor, which is known as PIGR receptors at the J chain region of the dimeric molecule. Now this complex of antibody A and the receptor is known as soluble antibody A or the secretory antibody A. Now this complex is easily taken up by the epithelial cells by endocytosis, and then it is transported to lumen in a vesicle. Once this complex in a vesicle reaches to the lumen, it undergo proteolytic cleavage, and the transmembrane and the cytoplasmic domain of this complex left attached to the epithelial cell, and the extracellular domain of the receptor which carries the antibody molecule is released into the lumen, and where these antibodies then neutralize the pathogen or the antigen. So this is basically how the whole gastrointestinal mucosal immunity works. Now the question is how the edible vaccines work. Now whenever we know that edible vaccines are to be consumed through oral route, so when we consume a transgenic fruit, say apple, it will enter into our gastrointestinal tract. Now since the antigen is enclosed in the plant cells, it will get protected by the acidic pH present in the stomach. So now they will transfer into the intestine, and intestine, when they reach, they will acted upon by the hydrolytic enzymes which will dissolve the plant cell wall and release the antigen into the lumen. And once the antigen is in the lumen, either it will be sensed by TLR receptors, or it will be transferred onto the dendritic cells, or it will be captured by the M cells and then transferred onto the dendritic cells. Now once it is being transferred onto the dendritic cells, these dendritic cells can activate move on to the lymph structures, which is Peyer's patches or the mesenteric lymph node, where they activate naive T cells to produce killer T cells and the regulatory T cells, and they will also activate naive B cells in order to produce the mature B cells or the memory B cells. Then these activated cells, they enter into systemic circulation. By the homing receptors, they reach to lamina propria region, and where these T killer cells will kill the infected host cells, because these infected host cells will act as reservoirs, so they will kill or they will remove those cells, whereas B cells will produce antibodies, and antibodies will neutralize the antigenic molecules present inside the cell, and some of the antibodies will be transported into the lumen, and they will neutralize that antigen there, and the memory B cells will retain that memory of that particular infection. So whenever in future, if the virus acts on the body, immediately those memory cells came into action and they will produce the antibodies. So this was a very I will say bird's eye view of the whole mechanism by which mucosal immunity works in GIT and how edible vaccines work. Now we move on to certain plants that are being used for developing edible vaccines. Now, there are certain characters of the plant that help in selecting a particular plant for the candidate for edible vaccines. Like one, the plant material or the fruit should be edible. It should be edible in the raw form, and it should not be cooked before it is being consumed, because cooking will expose that material towards heat, which will denature the antigenic molecule, or it will decrease the expression of that antigenic molecule. So whatever fruit we are using, it should be edible in the raw form. Then it can be easily grown. It can be easily grown in every season in every part of the globe, so that there is least transportation of that particular material. It should be compatible with the children's. Children can easily eat that particular fruit. So these were few characters that are used in order to select a particular plant for developing edible vaccines. So I will discuss few examples, like banana is one such fruit that has been used to develop vaccines for hepatitis B, for cholera, for HIV, and there are certain obvious advantages of banana, that it can be eaten in the raw form. No cooking is required, and children can easily eat it. But there are certain disadvantages also, that it has low expression of antigen. Low protein content is there, therefore low antigenic expression. Further, more than 3 years cycle of this plant, it creates a need for new transgenic fruiting plant to for the continuous supply. Further, then banana has a very low shelf life. You cannot store banana at room temperature for longer durations. Then we have potato, and potato has been used for developing edible vaccines for Norwalk virus, hepatitis B and cholera, and the obvious advantages are it's easy to store. It can be stored for longer duration at room temperature. And another advantage is that it is easy to grow. It can be grown in any part of the world. But the major disadvantage is that it cannot be eaten raw. There were few studies wherein the subjects were given raw potato to eat, the transgenic potatoes, but they exhibited certain side effects. Major ones were the diarrhea and vomiting, and moreover the raw potato was not palatable. So it has to be cooked before it is consumed, and cooking has shown to decrease the antigenic expression by 25 to 50%. Then we have tomato, and tomato has been used to develop anti-malarial and rabies vaccine, but the process for developing anti-HIV and anti-Alzheimer disease vaccine is still under process. Major advantages for using tomato is that it is easy to manipulate, it's easy to create transgenic potato, and it can be eaten raw. Further, new crop can be grown quickly. It has a very short cycle and you can easily grow a tomato plant very quickly. And the major disadvantages, again, it has a low shelf life and low protein content, therefore low antigenic expression. Besides these, acidic pH of tomato is sometimes incompatible with some of the antigens. They may get denatured in the acidic pH of the tomato. Then rice is another important plant that has been used to develop edible vaccines for hepatitis B, cholera and flu vaccine. Major advantages are that rice has a very long shelf life. You can store that rice for years, and rice is used in large quantities in baby food. So because these edible vaccines are being prepared with children as the major consumer, so since rice is being used largely in baby food, it can be used for vaccinizing or immunizing the children and even infants. Now the major disadvantage, we cannot eat rice in the raw form. It has to be cooked before it is being consumed. And cooking, as I told you, it may decrease the antigenic expression or the antigenic content by 25 to 50%. Then carrots are another important fruit that is being utilized for preparing edible vaccines, and anti-HIV, cholera and hepatitis B vaccines have already been prepared using carrots. Major advantages: long shelf life, can be eaten in the raw form, and children can also eat it because it's quite delicious in taste, and some children do like to eat carrot in the raw form. And disadvantages: it cannot be given to infants. Infants cannot chew the carrot, so you cannot vaccinize or immunize infants by having carrots as edible vaccines. Then lettuce is another example which has been used to develop cholera and hepatitis B vaccines, with major advantages that it can be grown easily and can be eaten in the raw form. No cooking is required. But the major disadvantage is it has a very low shelf life. You cannot store lettuce leaves for longer duration, and it's a seasonal crop in many countries. You cannot grow lettuce in India in any season, because you can grow lettuce in India only during the winter season. Now apart from these, there are various other fruits and vegetables like watermelon, peanuts, sweet potato, alfalfa, soya bean and spinach that have been used to develop edible vaccines. So friends, now we move on to the last aspect of this lecture series, that is limitation or challenges for these edible vaccines. Now, as we have discussed certain examples of the fruits and vegetables that have been used for preparing edible vaccines, but the selection of the fruits and vegetable is very limited. There are certain characters which I have briefly discussed, so one has to remain in the preview of those characters in order to select the plant or the fruit or the vegetable. Dose administration and calculation is very difficult. How much dose is going in one banana, how much dose is being administered by consuming one apple, it's very difficult to calculate. Then consistency of dosage from fruit to fruit may not be the same. So if one banana in one plant has good antigenic expression, another banana from the same plant may not have similar kind of antigenic expression. So that may lead to overexposure to the vaccine or underexposure to the vaccine. So overexposure may cause certain side effect, and underexposure may prevent the complete immunization of that particular person. Now it's very difficult to give to infants because infants may eat the whole fruit or may spit out some part of that particular fruit, which may again put a question on that how much dose has been administered to that particular infant. Some of the antigens or some antigens that are being incorporated in the fruits may get degraded in the stomach pH, though majority of the antigens are protected by the plant cell wall, which protect them from the acidic pH, but some antigens may be sensitive towards those pH and may get degraded into the acidic pH of the stomach. And last but not the least is the acceptability of the transgenic plant. So this becomes more or less an ethical issue, because a great portion of the population of the people still believe that these transgenic plants are not suitable for human consumption. So this is another challenge for the acceptability of edible vaccines. So friends, this was all about the edible vaccines, beginning from the definition of a vaccine, characters of a vaccine, types of vaccines, what are edible vaccines, how you can prepare edible vaccines, what are the basics of immunity, types of immunities, then basic mechanism of action of gastrointestinal mucosal immunity, how edible vaccines work, what are the different plants that can be used to develop edible vaccines, and in the last we discuss about the limitations and challenges for edible vaccines. Thank you.